Good morning everyone. So today Golgi body is one of the important cell organelle in cell. Golgi body. The Golgi body was discovered by Camillo Golgi in 1898. The size of this Golgi body approximately from size 0.5 micrometer to 1.0 micrometer. Generally this Golgi body has various names. So other names of Golgi bodies. Other names like it has several names like it also called in plant, especially in plant these Golgi bodies in plants, these Golgi bodies also called dictio, and also in various organisms like in yeast. Generally these Golgi bodies are extensively studied in yeast, hence in yeast this Golgi body is also called Baker's body, and also it has various names like lipo, and also another name for Golgi bodies Dalton's complex, or also we can use another name for Golgi bodies like idiosome, very important ideo, and we know that most commonly refer this Golgi bodies like Golgi complex, or also we can use the another term Golgi apparatus. Why means which is made up of with three different types of parts, also called Golgi apparatus. So these all are various names of Golgi bodies. Now we will see the structure of Golgi bodies. Generally this Golgi body has two faces. This Golgi body has two faces, like it has towards nucleus which it has cis phase, and towards plasma membrane it has trans phase. We will discuss how they are formed. So first cis phase and one more trans phase. Generally this how they are arrangements like this, for example, and yeah, here I am explaining, and this is the cell. Here center of this cell is nucleus, and here there are few structures which is arise like this. Yeah, so this is the structure of Golgi body, and in this Golgi body structure, here the structure which is present towards the nucleus means this one here. Yeah, this one. And there are some structures of Golgi body cisternae which is present towards the plasma membrane. See, this is our plasma membrane, this one nucleus. The Golgi body structures, especially cisternae which is present towards the plasma membrane, they are referred to as trans phase, and the cisternae of Golgi bodies which is present towards the nucleus, this one towards the nucleus, they are referred to as cis phase. Now we'll discuss what exactly this. Here generally this cis phase has convex in shape, very important, convex in shape, and they are proximal means near to the nucleus, proximal or near to the nucleus, and they are forming phase means first they are only first matured, sorry, not matured, it is a first to develop cisternae means they are forming phase. And very important, this cis phase present towards nucleus, very important, they are present towards nucleus, and this cis phase function very important role it play, that is which help it receive material from the endoplasmic reticulum, very important. Here I am writing this cis phase, it receives materials which is formed in endoplasmic reticulum like protein material or lipid material, and this cis phase of the Golgi bodies which receive those material of endoplasmic reticulum. So this is all about the cis phase of Golgi bodies. Now coming to trans phase. Yeah. Here trans phase, generally here the shape of this trans phase is differ, like it has a concave shape. Here I am writing concave shape, and it is distal means it is far away from the nucleus, means it is a distal or far away from nucleus. If proximal to the nucleus means cis phase, if distal or away from the nucleus means trans phase. And here they are maturing phase, they are mature cisternae of Golgi bodies, they are maturing phase, and very important, they are away from the nucleus, they are generally they are present away from the nucleus means it towards plasma membrane, very important, away from the nucleus or it towards the plasma membrane. So such structure is called trans phase. And what is the role of this trans phase means here I am writing here. So it alter and which ship the materials out. Generally cis phase which receive the material from endoplasmic reticulum, after receiving, then which undergoes, those materials undergoes various modification, then this trans phase which alter and which ship the materials out from the Golgi cisternae of Golgi bodies. So that is a major role of this trans phase. Okay, so here one important note, about cis and trans phase. Here cis and trans phases are non-identical. They are non-identical. Note, important note here, cis and trans phases are non, and they are structurally and also functionally different, but they are both phase, they are interconnected to each other, but they are inter to each other. Very important note here. Generally the cis and trans phases are non-identical in their structurally and their functionally, but they are interconnected to each other. Very important. Generally here, one important point here, sorry, here, yeah, these Golgi bodies generally present in organism. Here not all eukaryotic organism. Note here, note, not all eukaryotic organism contain these Golgi bodies. Only Golgi bodies present in eukaryotic, but not all eukaryotic. Why means this Golgi body is absent, exception, in some eukaryotic. Hence they are not all eukaryotic. Exception means they are absent in, this Golgi body is absent in human, which is absent in mature RBC cell, and they are in plant cell, which is absent in sieve tube cells of phloem. Sieve tube cells of phloem, in which these Golgi bodies are absent. Hence the statement here, Golgi bodies are present in eukaryotic cell, but not all eukaryotic cells. Why? Because these Golgi bodies are absent in some eukaryotic cells, like in human mature RBC, in plant sieve tube cells, in which Golgi bodies are absent. And now we move on components of Golgi bodies. Here, yes, components of Golgi body. Generally in Golgi body there are three components. There, first one, Golgi body cisternae. Second one is Golgi bodies vesicles. The third one is Golgi bodies tubules. How means? For example, here I will explain with the help of diagram. Here it is a plant cell or animal cell. Here the outermost layer, the plasma membrane or cell wall. In plant cell wall, it is the outermost layer. In animal cell, the outermost layer is plasma membrane. Okay, fine. So this is what? Nucleus. Center of the cell is called nucleus. So here how these components are arranged like this. Yes, here see towards nucleus. Yeah, so this is the structure of Golgi bodies. So in which this structure, and along with here there are some tubes like structure will form like this, tubules, and there are some vesicles like structures like this. Yes, so this whole structure is called Golgi bodies, and in which Golgi bodies there are three components. Now we will identify. So this flat disc shaped structure is called cisternae, and this spherical shaped structure is called vesicles, and this some tube like structure is called tubules. So likewise cisternae, vesicles and tubules, these three together called components of Golgi bodies. Now we will discuss each components in detail. Cisternae. Generally, this cisternae form from cisternae of endoplasmic reticulum, very important, which is formed from cysts of endoplasmic reticulum. And this Golgi body cisternae consist of many. Here there are many which consist of many flat or they are disc shaped structure. Generally, this disc shaped structure or flat structure like cisternae has only its diameter like 0.5 to 1.0 micrometer in length. Hence, we can use the complete size of the Golgi bodies is 0.5 to 1.0 micrometer. And coming to vesicles. Generally, these vesicles helps in formation of primary lysosome. Very important. Generally, lysosome formation occur from the Golgi bodies only. But these vesicles, in which component of the Golgi bodies helps in formation of vesicles? Lysosome means that is vesicles. So these vesicles helps in formation of, that is primary lysosome. Generally in lysosome we have different types. We will discuss in another video lecture. So these vesicles helps in formation of primary lysosome. Primary lysosomes. Now coming to tubules, and this is the tubules. Yes, yeah, okay. So this is all about the components of Golgi bodies. Very important point here. And if the cisternae of Golgi bodies faces towards the nucleus, they are called cis phase, and faces towards the plasma membrane or away from the nucleus, they are called trans phase. And in which Golgi bodies there are three components. One is, the most important component is cisternae, second one is vesicles and tubules, and these three components together called components of the Golgi bodies. Now we move on. Very important function of this Golgi bodies. Yeah. So function of Golgi bodies. Yes. Here first and most important function is formation of lysosome. Here the lysosome formation from vesicles of Golgi bodies. From just now we discussed from Golgi body vesicles. And second most important function, that is very important role it play in plant cell, that is for formation of cell wall. And one more, in animal, especially in human being, there is a formation of a membrane around the sperm, that is formation of acrosomes in animal or plant. In human being, sorry, formation of acro. So generally acrosome nothing but here. So in human or animal gamete, male gamete, that is sperm, if as like this structure means, and this head part of the sperm which made up of with a membrane like this, and this membrane is called acrosome membrane. So this acrosome membrane present around the head part of the sperm, and these acrosome formation are because of Golgi bodies. And most important, one more important function, that is processing and packaging of protein, very important here. And this Golgi bodies helps in processing and also packaging of materials, like the material which is formed in endoplasmic reticulums, like protein materials or lipid material. And processing and packaging of these materials helps in presence of Golgi bodies. Note here, very important note, Golgi bodies modify this protein or lipids. Important, this Golgi body modify only this Golgi body modifies this protein and lipid material, but it's synthesized by the rough endoplasmic reticulum and smooth endoplasmic reticulum. It means that Golgi body is not involved in formation of proteins and lipids. Just this Golgi bodies helps in modify the material which is formed from the Golgi bodies, endoplasmic reticulum. So it is important note here, Golgi body modify the protein and lipids which is formed in endoplasmic reticulum only. They are not helps in synthesis of proteins and lipids. And who responsible for protein and lipid synthesis means they are endoplasmic reticulum. Yes. And one more important functions of Golgi bodies, they helps in formation of glycoprotein and glycolipid. Very important. So they helps in formation of glycoprotein and glycolipids. Generally the formation of glycoprotein is called and here the formation of glycolipids is called glyco. Generally these glycoproteins and glycolipids are present in the plasma membrane. Generally how means, generally the plasma membrane structures if like this. So lipid bilayer plasma membrane structure we have like this, for example, and there is a protein structures. If it is a internal structure of plasma membrane means C, and this is a structure of what, lipids. If here glyco means glycogen or carbohydrate, if carbohydrate which attached to the lipid like this, or if glycogen or carbohydrate which is attached to the proteins. And here if this carbohydrate or glycogen which attached to this protein, then these together form glycoprotein structure. If glycogen or carbohydrate which attached to this lipid, then it is formed glycolipids. So generally the plasma membrane are made up of with glycoprotein and glycolipid in structures. How means, they form only in presence of Golgi bodies. So Golgi body helps in formation of this glycoprotein structures, this glycoprotein structure in plasma membrane, and also helps in formation of glycolipids in plasma membrane. So this is all about the formation of glycoproteins and glycolipids with by the process of glycosylation and glycosidations. And also very important role of this Golgi bodies, they also helps in, it is the only cell organelle, very important here note, it is the only cell organelles which show polarity. How means, this cell organelle shows two polarity. One is just now we discussed, that is cis phase polarity towards nucleus and trans phase polarity towards plasma membrane. So this is all about the entire concept of Golgi bodies. So here I have provided 3 MCQs from this topic here. The first MCQs, yeah. So consider the following statement regarding the structure of Golgi bodies, Golgi apparatus. Here Golgi apparatus. So there are 3 statements. So which of the statement given above are correct. Here identify the correct statement. One more MCQs here. Yeah. So read the following statement about the distinct phases of the Golgi bodies. Just now we discussed there are two phases, cis phase and trans phase. So read this three statement and which of the statement given above are correct. One more MCQs here. So which of the following statement correctly describe the metabolic association of Golgi? Here asking about function of Golgi apparatus. So here there are two options and select the correct answer using the choice below. So here, so that's it for today class. So if you like my video and hit the like icon and please share this video to your friends and don't forget to subscribe my Mahadyuth Academy channel. So thank you. Have a nice day.